All right. So chapter 20. Okay. Okay. So the microbial mechanisms of pathogenicity. Okay. 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 So talk about this stuff. Okay. So the first thing I want you to understand are the definitions. Okay. Again, these definitions become more important as we go along, right? So selective toxicity. So one of the ways that we have to deal with, or one of the things that we have to deal with, is to find a drug that ideally affects the host, or doesn't affect the host, but affects the pathogen. Okay. Um, Chemotherapy is by definition the use of chemicals used to treat a disease. Antibiotic is by definition a substance produced by a microbe that inhibits another microbe. Okay, antimicrobial drugs are by definition synthetic substances that interfere with the growth of microbes. Okay. Uh, so um, Alexander Fleming, in addition to discovering penicillin, 1932, this protozoal red dye used for streptococcal infections, 1940, first trials of penicillin. Okay, but in about 3 years, we went from having penicillin to having antibiotic resistance to penicillin. And if anything, the antibiotic resistance is going faster and faster and faster. Okay, so you can see here, okay, um, the these colonies here that are secreting things that are inhibiting other colonies. Okay, and you can see that there's a clear zone. That means that that substance is actually soluble in the agar and or therefore soluble in water. Okay. 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 Uh, gram positive rods. Okay. So these guys you don't need to know. Okay. So okay. Uh, so describe the problems of chemotherapy for viral, fungal, protozoal, uh, hemo, uh, hemophic infections. I can't say that properly. I'm sorry. And describe the terms here. Okay. Okay. So spectrum of activity basically is the amount of microbes it can actually influence. Okay. So narrow spectrum. Okay. Is of narrow range, like gram negative or gram positive, etc. A broad spectrum will affect a bigger range, but the problem with broad spectrum antibiotics is that your gut intestinal flora is bacteria as well, and therefore it makes you more susceptible to a super infection. Okay, a super infection is an overgrowth of normal microbiota. Okay, that can lead you to be as resistant to antibiotics. Okay, so if you kill off a large portion of your normal microbiota, then you can have things that are only supposed to be there in small amounts to over proliferate. Okay, like yeast infections are a good example of that. Okay. 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 So identify one reason it's difficult to target pathogenic virus because viruses live inside the host, so it's hard to kill the virus but not the host cells. Okay. While antibiotics with a very high broad spectrum, not as useful because you also destroy the gut flora. Okay, or the flora in other places. Okay. All right. This is definitely an important thing. Okay, identify the five modes of action of antimicrobial drugs. All right, so bactericidal and bacteriostatic. These are the definitions. Okay, so here, okay, make sure you're familiar with every one of these. Okay, so 1, 2, 3, 4, 5. You do not have to know um, the types of like uh, the name of the antibiotics that cause each type, with the exception of um, penicillin, chloramphenicol, and tetracycline. Uh, those are the three most common antibiotics, so I'm gonna hold you to those. Okay. Okay. Um, I think we talked about this first week or second week of microbiology already. Penicillin, okay, prevents the synthesis of peptidoglycan, so it interrupts cell wall synthesis. Okay, so it causes, if you can't repair the cell wall, it eventually will lyse open and like the bacteria will explode and it will leak out. Okay. Okay. Chloramphenicol and tetracycline target the bacterial 70S ribosome. Okay. The 70S ribosome is only found in bacteria, um, and therefore, uh, it you can inhibit that without inhibiting normal cellular or human cells. Okay. Okay. So you don't have to know this, right? But there's different places where you can influence um, the bacteria, the ribosomal bacterial ribosome. Okay. Okay. Injuring the plasma membrane. So the polypeptide antibiotics change the membrane permeability, makes them leakier, and therefore destroys them. Okay. Um, okay. Antifungal drugs. They combine with membrane sterols. So the fungal cell wall or the fungal cell membrane and the animal cell membrane are relatively similar. So we can't attack them the same way. But there's one thing that's unique to that the fungal cell membrane, and that is instead of cholesterol, they make something called ergosterol. Okay, and that compound you can attack because we don't have that. Okay, so again, the same thing occurs. You look for the differences between the two groups, and you attack one. Okay. Okay. So no. Okay. So basically, you damage the cell membrane, and that causes lysis. Okay. Okay. So inhibiting nucleic acid synthesis interfere with DNA replication and transcription. We do this with a lot of viruses, actually. Okay, inhibits the synthesis of essential metabolites. The sulfonamide, okay, competes with stopping the synthesis of folic acid. The microbes, a lot of them have to make their own folic acid, while we can actually eat folic acid. Okay. Okay.
Chemotherapeutic agents are antimicrobial drugs that fight disease by targeting a particular structural difference between human cells and those of the microbe. Because bacteria have many such differences, it's much easier to develop antibacterial drugs. Viruses depend on the host cell's machinery, so there are fewer differences to target. And eukaryotic pathogens, such as fungi and protozoa, have fewer differences from eukaryotic human cells, making it difficult to find drugs that have selective toxicity against these pathogens. Antibacterial drugs have several different modes of action. One of the ways in which antibacterial drugs can kill bacterial cells is to block the formation of bacterial cell walls. Normally, the peptidoglycan layers are held together with peptide crosslinks. Penicillin and its relatives block the formation of these peptide crosslinks. This weakens the cell wall, ultimately causing the cell to lyse. Antibacterial drugs such as polymyxin attach to phospholipids in the bacterial cytoplasmic membrane and interfere with its integrity. Eventually, the cell lyses. Because this mode of action also interferes with human cell membranes, this antibiotic is used only on the skin, where the outer layers of cells are already dead. Antibacterial drugs such as tetracycline, chloramphenicol, and streptomycin kill bacteria by inhibiting protein synthesis. Tetracyclines block the docking site of the transfer RNA, preventing amino acids from being added. Chloramphenicol prevents formation of peptide bonds between amino acids. Macrolides like erythromycin block the movement of messenger RNA through the ribosome. Aminoglycosides like streptomycin cause a change in the shape of the smaller 30S ribosome subunit, resulting in misreading of messenger RNA and insertion of incorrect amino acids. Bacteria have certain metabolic pathways that are not found in humans. One of these is the pathway for synthesis of folic acid, a coenzyme used in the synthesis of nucleic acids. Humans do not have such a pathway and require folic acid as a dietary supplement. One of the substrates in this pathway is para-aminobenzoic acid, or PABA. Antibiotics such as sulfonamides or sulfa drugs block a step in the folic acid synthesis pathway by acting as a competitive inhibitor to PABA. This prevents synthesis of folic acid and therefore synthesis of nucleic acids. The bacterium is unable to reproduce. Most organisms synthesize nucleic acids in a very similar manner, so it's much more difficult to find differences to target for these pathways. However, there is a group of synthetic drugs called quinolones and fluoroquinolones that block DNA gyrase in bacterial DNA. They have little effect on eukaryotic cells. Other drugs, such as rifampin, bind more readily to RNA polymerase in prokaryotes than eukaryotes and can be used to block transcription.
Okay. Actually, I'm gonna make sure you guys can hear me. Yes. So, screen. Okay. Okay. So let's go through the learning objectives. Okay. We'll skip this. All right. So one of the things you definitely have to understand is penicillin. The first, and it's still one of the most common antibiotics. Okay. And the first thing you have to know about penicillin is that it contains a beta-lactam ring. Okay. This beta-lactam ring is what makes penicillin useful. Okay. Uh, okay. But okay. So here is the beta lactam ring. So you can see that this area is like the ring, and this all is just kind of um, extra stuff. Okay, that makes it injectable versus oral, but basically they need this ring to function. Okay, we can modify penicillin. Okay, so we can take natural penicillin and modify it so it becomes less susceptible to antibody resistance. Okay, and keep the beta lactam ring. What things have what countermeasures other bacteria have done is they have made a beta lactam ACE. The beta lactam ACE cuts and destroys the beta lactam, so it no longer functions. And our counter to that is to make a either synthetic penicillin or a semi-synthetic penicillin that makes them resistant to the beta lactamases. Okay, so and the fight continues. There's always a fight back and forth. Okay, we can modify them, and then bacteria will evolve to counter that and be able to recognize a semi-synthetic penicillin and destroy that, and then we can modify it further, and then we can, so there's always a war going on. It's actually a very, very, very cool topic in biology. Okay, I can spend weeks talking about like different attacks and counterattacks and counter to that if I wanted to. It would bore you guys all to death, but I could. Okay. All right. So, so here is what happens. So they basically cut open the ring, okay, and that makes it no longer effective. Okay. All right. So there you go. So one of the things we do is make penicillin resistant penicillins. Uh, Also, we add beta lactamase inhibitors. So we can add, I don't even know how to say this, non-competitive inhibitor of penicillinase. That would block the penicillinase from happening. And the counter to that is they are evolving penicillinase that are that don't bind to this acid. Okay, so there's always a fight back. Okay. Okay, so more stuff. Okay, cephalosporins are similar to penicillins. They just have a different ring slightly. Okay, so it's a slightly different cut. Okay. So the angle of this is different. So the nitric it doesn't look like much of a difference, but it actually is when you look at the molecular structure of it all and the cut that it needs to make. Okay. We'll skip these guys. Hello, 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 hello. Okay. All right. Tell me my. Okay. Wait. Hello. Okay. Sorry, this has to be freaked out that I lost half of presentation. So I'm trying to not do that. Okay. Uh, okay. Okay, so this one should be obvious. This one is antibiotic resistance, right? So, okay. Inhibitors. Okay. Chloramphenicol, you should know. Okay. It binds the 70S ribosome, and therefore is broad spectrum because gram-positive and gram-negative bacteria still have the 70S. Like there's a lot of gram-negative bacteria that are not particularly sensitive to antibiotics or to beta-lactam stuff or the penicillins and stuff. Okay, but they are all sensitive to chloramphenicol. Okay, okay. They can suppress bone marrow, affect blood cell formation. Okay, so let me explain. Remember chapter 4 or 5 when I told you guys that mitochondria or just kidnapped bacteria and stuff like that? Well, kidnapped bacteria are still susceptible to the effects of chloramphenicol. Okay? Okay. Tetracycline also the same thing. Okay, they block the TRA attachment to the ribosome that prevents um, the the bacteria from making new proteins. So chloramphenicol and tetracycline, they are usually more bacteriostatic than bactericidal. Okay, they just prevent the bacteria. They don't kill the bacteria. They're not puncturing a hole in the cell wall. They're just making it not do anything. So it just stays around. Okay. Okay. So these guys will skip. Okay. These guys will skip. Okay. Okay. So polymyxin B and E, basically they're topical like bactericidals, and these are usually ointments that you rub and stuff. Okay. Okay. These guys you don't have to know. Okay. 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 So phthalamides. And so another one you do have to know. They inhibit folic acid synthesis. Okay. We don't have folic acid synthesis ourselves. We ingest our folic acid. So like bacteria, we can attack that pathway. Okay. All right. Antifungals. Okay. One of the key things about the antifungals we can do is attack the synthesis of ergosterol. Okay. Which makes the membrane leaky. We have cholesterol. So ours is different. Okay. All right. The rest of the stuff you don't really have to know. Okay. Okay. The reason why we kind of gloss over or I gloss over antivirals or antifungals is funguses don't usually cause serious disease. Most of the diseases caused by funguses are superficial. So, okay. Okay. Antiviral drugs. So, blocks entry or fusion inhibitors. Okay. If the virus can't get in, it's not functional. So, so you can either block the receptor, okay, on the host cell, or you can block the pathway fusion of the viral cell membrane or viral envelope and the cell membrane. Okay. You can prevent viral encoding. You can inhibit the viral DNA integration into the host genome. Okay, for certain viruses, you can have a nucleoside analog that inhibits DNA or RNA synthesis. So, what these things are things that look like A, G's, T's, and C's, but they're not. And so when the virus inserts them into the genome, it destroys the genome. Okay, and we can select for which ones attack to. Viruses have different RNA polymerases and DNA polymerases that we do, and therefore we can select for nucleoside analogs that the virus picks up, but we don't. Okay, and therefore we can attack that. Okay. Okay. So this is a cyclovir. Okay. See how it looks like G, but this part is different. This part, okay, it will try to insert this into the genome, but it won't work because it's missing this part, and therefore it will just stop, and that would prevent anything else from happening. Okay. Okay. So you can see like where it stops. Okay. Okay. Protease inhibitors. Okay. Protease inhibitors block the cleavage of the protein precursors, so they can't cleave themselves off, and that prevents the protein functioning normally. Okay. You can exit. You can create exit inhibitors. It inhibits neuraminidase, which is an enzyme required for viruses to bud from the cell, in particular flu viruses. Okay. Okay. Interferons obviously is an antiviral drug. So antiretrovirals, you can generate since reverse transcriptase is not natural to us, it's brought in by HIV, you can attack reverse transcriptase using drugs, and that is an antiretroviral drug. Okay. Okay. Protozoan drugs, you don't really have to know these. You don't have to know these either. They're harder to treat because they are actually animals, and so are we. So we're much more closely related to them. So we can't treat them as well. Okay. All right. All right. So this diffusion method. This is how we use this diffusion method. You can see there's a zone of inhibition. So like you can measure these with a ruler. For those of you guys in the lab, you guys have already seen this, but you can measure this with a ruler and see how big the zone is, et cetera, et cetera. Okay, and the bigger the zone, the more effective the antimicrobial. Okay. the other test is the E-test. Okay, you can measure the minimal inhibitory concentration. So you can see this tape right here has this drug in different concentrations, higher, top, and lower. Okay, and here you see the border where it's the minimal inhibitory concentration, and then below that they grow very well. Okay. Okay. We can skip the broth dilution tests. Okay. All right. So with every action we do to the bacteria, there is an equal or opposite reaction they do to us, right? So we invent a drug that kills them. They invent an enzyme that kills the kill switch. Okay. So here, okay.
A big challenge to medical science today is the growing problem of antibiotic resistance. A pathogen that is resistant to a particular antibiotic will not be killed or inhibited by that antibiotic. It has gained that resistance either by random mutation or through gene transfer from another bacterium. In a particular population of bacteria, there may be a few cells that are resistant to the effects of a particular antibiotic. When the patient is given a particular antibiotic, that drug will kill or inhibit the growth of all the sensitive bacterial cells. This allows the resistant cells to multiply freely in this less crowded environment. One way that bacteria can acquire resistance genes is through mutation. If a mutation occurs in a sequence of DNA, this could result in a different amino acid sequence. Consequently, this different sequence could produce a new enzyme, perhaps one that can interfere with the action of an antibiotic. Bacteria can also acquire resistance genes by horizontal gene transfer. One type of horizontal transfer is transformation, in which a cell picks up a piece of DNA from its surroundings. Or a bacteriophage can transfer the resistance genes from one bacterial cell to another in the process of transduction. Finally, resistance genes can often be found on small pieces of DNA called R plasmids or R factors. These R factors are often transferred via conjugation between bacterial cells.
All right. You guys don't know about this already. Okay. Hey, let's see. Okay, cool. I just want to make sure I can be heard. All right. Okay. The mechanism of resistance, another important page. Examine destruction or reactivation of the drug, prevention of the penetration to the target site within the microbe, alteration of the drug target site, the rapid efflux, okay, of the antibiotic or some combination thereof. So another important slide right here. You can either prevent the antibiotic from coming in, you can get rid of it as soon as it comes in, you can modify your DNA or your genes that the target is no longer matched to the antibiotic, or you can just destroy the antibiotic as soon as it comes in. Okay. Okay, so you can see here, so initiation of antibiotic therapy, the immediate bacteria count drops, but you have some that survive. And then what happens, okay, is the count goes back up. Okay, so you can see the amount of antibiotic needed to control growth. It goes up higher and higher, and eventually it doesn't control growth at all. Okay.
Bacteria can resist the effects of antibiotics through several mechanisms. They may secrete
Oops. Uh, okay. So, screen, oh, check. Oops, okay.
Bacteria can resist the effects of antibiotics through several mechanisms. They may secrete enzymes that destroy the chemical structure of the antibiotic. For example, penicillin-resistant bacteria produce an enzyme called beta-lactamase. The beta-lactamase enzyme breaks the beta-lactam ring in the molecule of penicillin and related antibiotics, such as cephalosporins. This prevents the antibiotic from disrupting the peptidoglycan cell wall structure. Other bacteria prevent antibiotics such as tetracycline from passing through their cell membranes. They do this by changing the structure or charge of pore proteins. Even if the antibiotic can penetrate the cell, a resistant bacterium can make it impossible or more difficult for the antibiotic to bind to its target. Sulfonamides are antibiotics that compete successfully with PABA for an enzyme binding site in the folic acid synthesis pathway. Resistant bacteria may have evolved an enzyme with greater affinity for PABA, thus making the sulfonamide less effective at blocking the folic acid pathway. Other resistant bacteria have developed a method to obtain molecules they normally synthesize directly from their environment. Therefore, they aren't affected by the blockage of the biosynthesis pathway. Finally, some resistant bacteria have membrane-localized efflux pumps that remove some antibiotics from the cell. Some resistant bacteria have a greater number of efflux pumps, thus increasing their ability to eliminate the antibiotic. In some cases, bacteria have also been able to increase the spectrum of drugs that are eliminated by these pumps by making pumps less specific.
All right. So, okay. So, this is why you shouldn't do any of these things. Okay. Or using antibiotics for the common cold. I hate that. Using outdated or weakened antibiotics, that generally builds up resistance. Okay. The biggest problem right now actually is the number 3 here. We're using more and more antibiotics in animal feed, and they're becoming antibiotic resistant strains in animals that will then be spread to humans. Okay. Okay. So the therapeutic index, okay, is the risk versus benefit. So the first number is the minimal amount that, or the minimal amount that can generate risk versus the amount that can generate benefit. And so the bigger the difference between risk and benefit, the more useful it is. Okay. Or the top number is the maximum amount that you can take before there's risk. The the bottom number is the minimal amount that there is a benefit. And the bigger the difference between the two is the therapeutic index. And that the higher the therapeutic index, the better. Okay. Okay. So synergism is basically when it affects two drugs together is far greater than the one each of them alone. So in mathematical terms, is 2 plus 2 is equal to 5, 6, or 10. Okay. Antagonism is where they get in each other's way. So in mathematical terms, 2 plus 2 equals to 2 or even 1. Okay. And so here you can see the effects of synergism. So there, this circle, this middle region right here, you can see there's both drugs, and that actually has effects of increasing the zone of inhibition. Okay. Okay. Don't worry about these guys. Okay. All right. So hopefully you understood that chapter. All right. So see you guys for chapter 21.